Welcome everyone. My name is Susie O'Hara and I'm the project curator for the Human Cell Atlas UK Public Engagement Programme, which is funded by Wellcome Trust and entitled One Cell at a Time, bringing together communities, patients and researchers to build the Human Cell Atlas. I'm absolutely delighted to be joined today by our project in collaboration with the Royal College of Art, Esther Teichmann, Christopher Stewart, Boris Jardine, and Sarah Teichmann, who from the Human Cell Atlas project. Our back of house team is Tracy, Charlotte and Paul, who will be helping with the technological side of things, and we'll be thanking them no doubt later. The aim of our project is to bring about a cultural shift towards enhanced and sustained engagement with the Human Cell Atlas across research and public communities. And our project kind of sits within the nexus of art and science. Our public engagement activities are underpinned by core questions that have emerged from and could inform the science of Human Cell Atlas. The first question being, what is it to be normal? The second, what influences people's value and trust in research involving tissue donation and open access data? Our aims within the programme, which will happen across 2021, is to build a culture within the HCA where UK and international members seek out and value the opinions and experiences of patients and communities who donate to our research studies. We want to co-develop a common language that enables HCA members to effectively communicate their work to different communities. We want to normalise conversations within families and communities around tissue donation for research. And we want to generate dialogue and transparency between the HCA and the communities that the communities that we engage that address questions and concerns around our research, particularly in relation to tissue donation. The ways in which we've chosen to do this is to work in collaboration with a wide and broad diverse range of artists coming from different disciplines, using different tools, different methodologies and bringing different perspectives to these core questions. We've commissioned 10 artists across the UK to respond and work with communities and HCA members to explore and interrogate the questions that we've laid out. We'll be working with schools in partnership with Hayes Design and Little Inventors, and I'm proud to say that we're going to be launching our How to Build a Human Card Game Challenge on the 8th of January 2021. We've just launched this week our Young Poets Challenge, the language of cells, poetic science, and we've also generated a bite-sized animation exploring how the Human Cell Atlas is helping to combat COVID-19. All of these projects can be viewed on our public engagement webpage on the Human Cell Atlas website. But we're here to talk about the Human Cell Atlas and the technologies of seeing for our second ArtSci Salon. During the project, during this event, we'll be using Slido.com to facilitate your questions, and we hope that you bring out your mobile phone now, go to Slido.com, type in ArtSci 02, where you can then ask questions and upvote the ones that you like. You can stay anonymous or you can attach your name to the question. And we'll be delivering live polls to make sure that you're keeping engaged. So we're going to try doing that now. So Charlotte, if you can change the screen to the Slido poll. Our question is, before today's event, have you taken part in any other HCA public engagement event or activities? So Charlotte is sharing her screen now. And we have takeoff. So 100% of people who are joining us at the moment. Oh, it's gone down to 80. So we'll just let this continue just for a couple of more minutes. And while I tell you that the schedule for today's event is we're kicking off with Sarah Teichmann and Esther Teichmann, who will introduce the speakers a little bit more formally, and then this will be followed by a discussion, a live Q&A between Esther and Sarah. And this will be then followed by a series of presentations by Boris Jardine, Christopher Stewart and Esther Teichmann. And at the end of the session, we'll have space and time for your questions using the Slido platform. So I do hope you enjoy. So we have a 53% yes and a 47%. This is the first time that you're here. Very, very welcome. I'm delighted everybody could make it. And so with that, I'll pass over to Esther. Thank you very much.
Thank you so much, Susie, and brilliant to have the whole project overview. Thank you to all our speakers for joining us today, to Boris, Chris, and Sarah. And I'll introduce our speakers one after each other just before their talks. But I wanted to kick off today's event with just a few introductory questions to Sarah. Sarah Teichmann is the co-founder and co-leader of the Human Cell Atlas International Consortium and is head of cellular genetics at the Wellcome Sanger Institute. And she's also my big sister. And the way Chris and I came to know about the Human Cell Atlas project was many years ago, before it was called, it had a name. And we were really fascinated by Sarah's initial descriptions of the project and early ideas, and all of the intersecting narratives that kind of began to spiral from early descriptions of what the HCA might be and could be and has become. So Sarah, thank you for joining us. And thank you, Boris, for helping me formulate some of the questions that would be a good introduction to today's session with Sarah. So Sarah, we wanted to ask you a little bit about the origins of the HCA and whether you could tell us about how and when you first thought about the possibility of the project and what form these initial ideas took.
Sure, thank you, Esther, and thank you, Susie, for having me at this event. So the origins of the Human Cell Atlas project are really in the development of single-cell genomics technologies, from my perspective, and that those go back to kind of over 10 years ago now, when it first became possible to sequence the nucleic acid content of a single cell. Because prior to that, genomics really relied on bulk DNA, RNA, or open chromatin sequencing, which required hundreds or thousands of cells, so a lot of material. And then it was the miniaturization of the technology down to the content of a single cell, that resolution revolution in genomics, that catalyzed the idea of expanding that up and studying all the cells in the human body. And here in the UK, on the Genome Campus, I started using single-cell genomics technologies and applying them as already when I was at the MRC laboratory of molecular biology, and then when I moved to the Genome Campus to a joint position between the EBI and Sanger, I co-founded with other faculty members from those institutes a single-cell genomics center, and it was during my move and my discussions with Mike Stratt in particular, that we started, considering the idea of scaling up these technologies to profile an entire human. And then over time, the idea kind of grew into including spatial technologies. I connected with Aviv in 2016, and then the reality of an international community to tackle this, made a lot of sense. Basically, it was like a logical next step. And Aviv coined the term Human Cell Atlas. Similar ideas were bubbling up in different places around the world at that time, and we got together to launch the initiative, and the rest is history, really.
Thanks. No, that's really great to have that insight. And you started to touch upon the naming of the project, and that Aviva Grev, who's the co-lead of the project with Sarah, coined the term. But we were wondering if there were in that naming, whether there were other names that were considered. I know you spoke about your initial thinking of the project as a Google Maps for the body. And I was wondering, before you kind of settled on Human Cell Atlas, were you thinking about terms like library, encyclopedia, database, or even project, as in the Human Genome Project, that maybe can be thought about as a central precursor to this? If not, that's fine.
Yeah, so the word, for the sequencing of the human genome, it was called the Human Genome Project, and certainly, libraries or encyclopedias are really suitable analogies as well, for sure, and kind of capture different elements of the way we're trying to catalog ourselves. I think what the word atlas or map adds is that spatial dimension of a coordinate system in 2 and 3 dimensions. And a human body is a 3-dimensional entity. It's so is a DNA molecule when it's wrapped up in chromatin inside our nucleus. But it's the information is really encoded in that linear sequence. So it is a list of the bases. Whereas for the human cell atlas, it is atlasing or mapping the cells as they sit inside our tissue. And so capturing that 3-dimensional notion, I think, is the word atlas is really nice in that way.
And we were also wondering, and Boris particularly led us to this, whether the HCA relates to other projects and how you've thought about those relationships, or if, for example, ENCODE, the Encyclopedia of DNA Elements, or projects in different disciplines that may have provided inspirations or models. Were you looking to other projects in terms of maybe the structure or other elements, in terms of thinking about, similar projects either in the present or historically?
Well, the human cell atlas definitely relates to precursor projects that lay the foundation for it, both the Human Genome Project, ENCODE, more specific projects like the brain cell census and so on. I mean, and it relates to those projects not only in terms of the actual data and information. I mean, there would be no human cell atlas without the human genome sequence, so without the Human Genome Project. It also, so it definitely builds on, we stand on the shoulders of giants. It builds on all of those projects for sure. It has synergistic relationships with data from other projects like the Human Protein Atlas that's been going on for 20 years and sort of mapping individual proteins in human tissues. And in terms of the kind of model for how the scientists are organized, certainly the Human Genome Project was the precursor of consortia in biology. I mean, we've also, at one of our human cell atlas meetings, we also had a guest speaker from CERN, and that was, a post-World War II European project for high energy physics and particle physics, and that, they've organized themselves in a different way. The Human Genome Project was again organized in a slightly different way, the International Cancer Genomics Consortium, but they're all, what they have in common is that they're communities of scientists, many scientists working together to achieve a common aim. And in Human Cell Atlas, that's essentially what we're doing. There are over 2,000 members from all over the world. We're all working together to achieve a common aim. And I guess we're trying to do that in a very modern, democratic, grassroots way. I guess that's maybe the novel dimension there.
And our final question touches or centers around that relationship to community and the network of the scientists that comprise the HCA. And I know you and I had spoken a little bit about the unexpected aspects that have emerged from the project and that have become especially apparent within the COVID research. And I was wondering if you could tell us a little bit more about these recent aspects of the project that were unforeseen at the start, the power and the kind of the functions that this network has that maybe weren't quite imagined to start with.
Yeah, certainly the community pivoting towards COVID-19, has been quite an amazing experience, and the agility and speed with which the community rallied to map the viral entry receptors across human tissues in the COVID-19 Cell Atlas project was pretty amazing. And that, I think that was possible because we had a pre-formed community. It also, it was only possible because we had that sort of understanding and that structure in the first place. And at the same time, collaborating on mapping the viral entry receptors and covariates with age and gender and so on, that also, it strengthened the community in itself. It catalyzed and accelerated the formation of the biological networks, starting off with the lung biological network. And so it was a sort of a positive feedback, in a way, a virtuous cycle of having a community, and the community became strengthened by really focusing and having minds sharpened to respond very, very quickly to the public health emergency. And that's the video that Susie mentioned in the previous slide, kind of refers to that project.
Thanks so much for joining us, Sarah. Susie has one final question for you before we let you go. Over to you, Susie.
Thank you so much, Esther. Sarah, it's amazing to listen to the kind of background thinking that's gone on around developing the Human Cell Atlas project. And I'm genuinely kind of impressed and, you know, really intrigued by the interdisciplinarity that is really at the core of the HCA project. And from my perspective, obviously from the public engagement and bringing artists in as collaborators in this project, I was just really interested to know kind of from your perspective, the value of adding in the kind of artistic kind of tools, perspectives, methodologies into this mix of knowledge generation that the Human Cell Atlas is facilitating internationally.
Well, you're right that the Human Cell Atlas is an interdisciplinary project, kind of, and it always has been from the very get-go. Because, as you know, it was founded by two computational or biologists. We sort of kicked it off, but it's by its very nature, it involves the biomedical and clinical community, as well as the genome technology community. And really, you know, expanding to science is kind of natural. Expanding to art, you know, connecting the science to the art is just another step in a certain sense. I think that the value for the scientists is, you know, you know, it's in many different dimensions. I guess it gives us more of an awareness of sort of issues, applications, impacts, and so on that our science has more broadly, kind of in society. And, you know, makes us sensitive to you know, to things like details of wording. You know, Esther just mentioned why do you use the word atlas? I never really thought about it kind of in detail. So I think it's you know, it's fantastic, and thank you so much for, you know, helping us achieve this this dialogue between science and art. It's you know, it's an incredible project, an amazing opportunity, and for the Human Cell Atlas. So thank you very much.
Goodness, it's an absolute pleasure and an honor, Sarah. I'm really interested to kind of throw it back out to our audiences now with another live poll that asks the question, which city are you zooming in from? It would be really interesting to know how the breadth and spread of our audience today. So Charlotte, can you maybe share your screen? And I invite people to look on slido.com and share where they're calling in from with us. We've got Cambridge, Edinburgh, London. I'm in Newcastle upon Tyne. I think we have London. Cambridge is Boris. Sarah, are you Cambridge?
Sarah is Cambridge.
Yes, indeed. Yeah. Yep, I am.
Fantastic. So we've got some here. Wow. A really nice spread. Bury St Edmunds, Edinburgh, Bristol, Asheville in the US. Fantastic. Amazing. Really wonderful. Thank you so much, Charlotte. Please do continue sharing with us on the Slido. But without further ado, I'm absolutely delighted to welcome Boris Jardine to share his screen and do his presentation. Thank you, Boris.
Thank you so much, Susie. I'll give a brief introduction to Boris. So Chris and I are working on an artist film. We're just in the first very few weeks of the project, really, although we've been thinking about it for several years now. And our artist film is working with the narratives around the HCA, and we're thinking about the process and the movement from flesh to data. And we were really excited to use the opportunity to collaborate with creatives and researchers and academics from other disciplines within our small commission. And we're working with the composer Deirdre Gribben, who I have worked with previously and who Sarah has worked with as well, who's been a Leverhulme Fellow at the Sanger. And we're really, really excited that Boris took up our invitation to research and write an overarching contextualizing piece on the Human Cell Atlas that touches on the different strands and interconnected histories. So today Boris will be looking at the history of the scientific atlas, but other themes that he's exploring later on in this research piece is looking at, for example, the open data movement and post-war developments of big science, and looking at previous examples such as the Human Genome Project, as well as thinking about the individual and the collective, how we move from the body, from the flesh to data. And today he'll be exploring the origins of the term atlas and outlining its histories and mapping these for us. Boris is an academic who's based in the, sorry, I've just lost my thread, based in the History and Philosophy of Science and Medicine department in Cambridge. And Boris's research deals with the history of scientific instruments and the material cultures of science. His current research project looks at the lost museums of Cambridge science, 1865 to 1936, and tells the story of the new museum site in the center of Cambridge, focusing on the way in which collections were amassed and then dispersed in various museums. And Jardine is also working on a book entitled The Plans for Utopia, Modernism and the Sciences in Interwar Britain, that come out of his PhD research. He's previously been Curator of Science at the Science Museum and Mumby Fellow in Bibliography at the Cambridge University Library. So thank you so much, Boris, for working with us and for joining us today.
That's brilliant. Well, thank you very much for inviting me. I assume that you can hear me and see my screen. Is that right? Great. Okay, that was a really good and useful introduction. So I don't need to say too much about who I am and what I do. It's been really exciting working on this already. I should say that we're just really at the beginning of this research process. So this is kind of work in progress, and also, as Susie was saying, this is part one of a three-part project. So as Esther's just said, I work mainly on the history of scientific instruments. In fact, that's kind of my main focus, and I've done a lot of work on early modern instruments from the 16th to 17th century. But I have quite diverse research interests, as is often the case when people like me move between academia and the museum world. So that probably explains why I work on so many different things. The project that's probably most relevant to this was called, let me just bring up my second slide, was called the Total Archive. So this was a project that I came up with a group of colleagues in around 2014-15. This is an image of the now demolished Cincinnati Public Library. And the Total Archive, as we called it, was an interdisciplinary research project looking at the history of attempts to create systematic collections of data or information. And it was an interdisciplinary project. We had a conference at Cambridge's Interdisciplinary Research Centre, Crash, in 2015, that brought in artists, historians of art, economic historians, historians of science. And we really looked there at a broad range of things like encyclopedias, libraries, and more closely focused research projects that had happened in the 20th century, especially in the human and biomedical sciences. So we actually had a talk on the Human Genome Project at the conference. This resulted in a couple of publications. This is the cover of a special issue of the magazine Limb, which is available, that's open access online. That contained a kind of a selection of papers mainly drawn from the conference. And there was also a special issue of the journal History of the Human Sciences, where we really zoomed in on the 20th century history of total archives, as we called them, with quite an unusual, and this is kind of relevant, kind of unusual array of areas of research and study and information gathering. There was a paper on an attempt to collect data on human dance from around the world. I wrote a paper about a thing called mass observation, which is an organization in the 1930s that tried to collect everyday kind of anecdotal evidence about people's opinions, beliefs from across Britain, and they thought the world, it was supposed to be a kind of dynamic archive of opinion and prejudice and belief, partly as a political instrument, but also as a social scientific research tool. So that's kind of and I think as you'll see, that there are multiple reasons why the Human Cell Atlas is relevant here, mainly because of its attempt to be comprehensive with regard to one specific domain, i.e. the cells in the human body. But that's the lens that I really look at this through. So, but the question about how to historicize and how I'm going to historicize the Human Cell Atlas is a really big and difficult one. It's one that I've been grappling with as I start the project. One way of doing it, which is different from what I've ended up with and where I'm going, is, but I want to acknowledge, is the history of this Human Cell Atlas that's kind of embedded in the project that we've already had quite a good summary of from Sarah. This is an image that features in the 2017 white paper that really sets the ball rolling with the HCA. This is quite a famous illustration. It is where the term cell is coined in terms of biology. This is from Robert Hooke's Micrographia 1665 with an illustration of a thin section of cork. And this is part of the history that is told within the HCA, and that is a narrative of the development of cell biology, mainly in the 19th century, and then attempts to create reference collections, classifications, typologies of cells, and then specifically attempts to create systematic collections, reference collections of human tissue samples. And this is a really important history, and it's actually part, I think, in a really crucial way of the science of the HCA itself. It's a way of legitimating, contextualizing, and giving meaning to the work that's done. And obviously, in a more local and simple sense, the science simply draws on previous work, as Sarah said. The HCA wouldn't exist without the Human Genome Project. So that's one approach, which is kind of, I suppose, the main through line through the history of science to the HCA. But I'm actually trying to do something different. So I wanted to come up with something that also would be useful to Esther and the work that she's doing with Chris on the film, and that would give a kind of different perspective on the HCA. And I started to think about something the HCA does itself, which is to think about scale. So we've already heard that the HCA is often described as a kind of Google Earth or Google Maps for the human body. And I started to wonder if this question of scale might be interesting in terms of historiography as well. So I won't give too much background on this, but the question of scale in the history of science is a really important one. There's been a big debate over whether we should and can legitimately tell really long histories of science. They're kind of what's called in my discipline Plato to NATO view of history, histories of the scientific revolution, histories that go from the 16th century right up to the 20th century. And more recently, there's been a very strong trend towards what's called microhistory, so extremely closely focused research into specific practices of disciplines or even individual laboratories at a certain time. And there are challenges to that and questions about which one is valid and how they relate together. But I thought this was a useful opportunity to kind of, in a way, revisit that question in a sort of slightly soft touch and maybe quite creative way. So the top image that you can see there is part of one of the ways that scale happens or scaling happens in the HCA. You can see a kind of zooming in through RNA analysis to specific clusters, and then a closer focus, and then a kind of individual analysis using this single cell RNA study that they do. And there are lots of different kinds of zooming as well, right out to whole organs, and then out to the whole body, the location in the body, and back down to the individual cell. So I thought, historians always like to think in threes. So I thought, is there a kind of 3-stage history of the HCA that I could provide? Could there be, for example, a kind of macro, a meso, and a micro history? And that's what I've come up with on the bottom here. So you can see on the bottom left, this is, I'll come back to it in a second. This is an atlas image. This is an atlas in the sense of mapping, so it's the most obvious meaning of the term. And this is really a history that dates from the 16th, 17th, and 18th century. to the history of scientific atlases. That's what I'm going to be talking about today. Second image comes from one of the many community tools developed in around the middle of the 20th century to do with the Drosophila fruit fly genetics community. And this is the kind of middle scale that Sarah's already touched on, but this is really about the open science movement, data sharing practices in the life sciences, the idea of, is there such a thing as big biology? We think of big physics and big science and huge scientific instruments and experiments, but can we also think of distributed communities of scientists working on a common problem as a form of big science, in the life sciences especially? And there's an interesting continuity here with the atlas question, where what are the tools, especially the visual tools like this map of a fruit fly, which then allows you to key into the all of the genes and the mutants that exist in fruit flies that you could then send off for and get in the post. Then there's the micro history. This is in some ways the most difficult and the most complex to approach, kind of oddly, given that it's the most closely focused. The image on the right is from a paper in Nature Neuroscience, in which a group of scientists used RNA analysis to identify a new, or at least to kind of bring to light, a new kind of neuron. And that's the kind of work that the Human Cell Atlas specializes in. This is really zooming into one specific community of researchers working on one specific part of the human body. And also in this image, you can see the relationship between the tissue sample and then the data. And so it's a question of how tissue is handled, how samples are identified, or individuals that you want to collect samples from are identified, and then how that kind of physical, wet laboratory work goes into this kind of dry numerical data science, and how you retain a kind of visual language for those things. That's the third part. So going back to the first, I want to just give you now a very brief kind of overview of this idea of the history of a scientific atlas, this kind of macro, zoomed out view of the HCA. In some ways, it's quite a surprising story, because atlas has this spatial sense we've already heard, and we can all think of what an atlas is, but it's quite a nice history, and it's one that's possible to write, mainly because of this book, which came out in 2007, which I'd strongly recommend. It's a really wonderful book. It's been important in the history of science, but it's also it's a visual history of science. And the title doesn't convey that much. It's called Objectivity by Lorraine Dast and Peter Gallison. But it's really a history of scientific atlases. And they use this figure of the collection of images of science in order to chart attitudes towards truth to nature. Well, they create this succession of versions of objectivity, truth to nature, mechanical objectivity, and so on. There's a nice quote that I pulled, which defines the atlas as a systematic compilation of working objects. Atlases are the dictionaries of the sciences of the eye. That's from the first section, where they really give this overview history, which I'm in some ways paraphrasing and also going into a little more detail on. So the first atlas, unsurprisingly, is an atlas. But of course, this is a slightly strange name to give to a collection of maps. This is the 1595 cosmographical atlas by Gerard Mercator. Mercator is famous for the Mercator projection, the first kind of modern map, really developed for navigation, and slightly before this, but begins the tradition of the use of projective geometry in modern map making. He decided in 1595 to call his collection of maps an atlas. So it's already a metaphor, because of course an atlas is the figure from ancient mythology, the giant who was burdened by having the world put on his back. So there's in a way, quite a literal kind of carryover there. I'll show you this wonderful image, more or less contemporary with Mercator. This is an image of Atlas with the world on his back, but there's also an artistic and a scientific image. This is by Peter Paul Rubens, engraved after Rubens' drawing for Francois d'Aguilon's book on optics, published in 1613. Optics, of course, is a science that's closely related to developments in Renaissance art. And this is Atlas carrying the celestial sphere and really illustrating how projection works, so how the map is drawn effectively. So it's a really nice kind of infolded image. Obviously, Mercator's map was extremely successful. It set the standard for map making, and his atlas became the standard term for collections of maps a little bit later, so from the 1620s and 30s. And that obviously continues to this day. But then there's another metaphor. So you've got the metaphor of the from Greek mythology, and then you've got the second metaphor, which is what other kinds of atlas might there be? And rather wonderfully, the first atlas in this metaphorical sense is a collection put together in the 1650s of icons of the Virgin Mary collected from around the world. So this is a project by this guy Wilhelm Gumpenberg. It's called the Atlas Marianus. It was published in 1657, and he was a Jesuit, and his project here was to defend the use of and really the cults surrounding images of the Virgin Mary. So it might seem like a kind of random thing that this collection gets put together, but in a way it's not. I mean, who better than a Jesuit to defend the idea of a collection of images as this validation of a whole part of religious practice. So these are images that were copied from shrines around the world and then collected together. There are hundreds of them. This is obviously a collection of 7, and then the frontispiece just on the top left there, you can see it. That's the Ascension of the Virgin Mary. So this is the first atlas in the non-mapping sense, although it obviously has this cartographic feature of being a global survey. Relatively quickly, you have this transfer. I mean, I'll get into the question of what you can call an atlas and what you can't, but the first real use of the term atlas in the sciences is this, as far as I can work out. This is Tobias Mayer's Mathematische Atlas of 1745. So quite a long time later, but it's kind of complicated. The idea here is that he's created an atlas of the mathematical sciences, and what he means is a collection of plates. So this is large images showing all of the different parts of mathematics. This is not mathematics in our sense, pure mathematics. This is the applications of maths to, or the mathematical sciences to astronomy, surveying, fortification, and so on. So it's kind of the world of mathematics, I suppose you could call it. But there's something interesting here. So an atlas is a systematic collection of images. You can kind of get that. But the reason that you get this thing called a scientific atlas is actually slightly different. And that's that, and it's really a media question. So I'll show you another image of an atlas. This is Benjamin Wilkes' collection of images of the English moths and butterflies. This is an atlas not because of its title, but because it's printed on atlas paper. So atlas paper is a specific format, a large format and high quality paper that's in use in the 18th century. I don't know if it was in use before, but by the 18th century, if you want to print something that's a collection of plates, if you're well enough funded, you'll choose atlas paper. And here's a point that Daston and Galison make, that really once you have systematic collections in large format, these are atlases in the kind of strong sense that they're physically atlases. And here we have the development of the scientific atlas proper, really starting in the late 18th century, mainly with atlases of anatomy and soon after pathology. That's a kind of interesting comparison just initially there. But I want to just focus on this one briefly. This is Albinus's Tabulai Scaletti of 1749. This is a really stunning atlas of human anatomy. It took maybe a decade to create. There was an articulated skeleton that he was working from. He had an engraver kind of on hand to make these. And he actually, a rather disturbing story, employed an extremely thin man to take his clothes off and stand in the poses so that he could then match the skeleton to the physical form. So these are, in a sense, kind of from the life, but just of the human skeletal structure. But there, again, Daston and Galison make this point. These are idealized images. So this is a kind of perfected skeleton. And that's really the whole style of these late 18th century atlases. In the 19th century, the term becomes generalized. You have atlases of all sorts of things, and the atlas volume just becomes the plate volume as a component to a scientific text that's systematic with regard to some branch of science. And here you have this great flourishing of atlases that continues right into the 20th century, and with the Human Cell Atlas continues today. So I'll just give you a kind of glimpse of the wonders of the scientific atlas. The top left, we've got a 20th century atlas of colors. On the right, an atlas of snowflakes from 1913. And then the bottom left, quite a famous one, the Atlas of Clouds, which was published in 1896. So it's probably worth just pausing here and maybe also wrapping up. What's the significance of these atlases? I mean, obviously they're attractive objects. They're now, extremely valuable books that you can buy at auction. They're full of these really wonderful plates. Daston and Gallison give this extraordinary history of the techniques, lithograph, mezzotint, photomechanical reproduction that are used to create these standard objects. But what's the importance of them? They're really central to scientific endeavor. That's the thesis of Objectivity, the book. At the most basic level, they define objects of inquiry. So if you want to make an object central to a branch of scientific work, you create an atlas of it. And that can happen with completely novel things, like, for example, when you start to be able, with the cloud atlas in the early 20th century, to create the tracks of atom particles from radioactive materials. You immediately have to create an atlas of the decay patterns of all the different substances that you want to look at. So it's a way of, in a way, bringing into being, for scientists outside of one laboratory, an object of inquiry. And the cloud atlas is a really wonderful example of this. The clouds are classified and named in the early 19th century, but it's really only with, and that's the famous moment in the history of meteorology, with Luke Howard naming the clouds, but they only become scientific objects in a meaningful sense at the end of the 19th century, because it's only with the cloud atlases that were agreed upon at these massive international congresses in meteorology that people around the world are able to say, well, I know the name, but I'm also looking at the same cloud as you are. There's no way of doing that without an atlas, because obviously the forms are fleeting, and you can train yourself to a certain degree, but you need a visual reference. So this is why atlases define the normal or the typical or the standard, and that's a really important function. I think it's a really important function in the HCA, and it's always been an important function. This is particularly important in pathology. I think that's one of the main areas where you have obviously atlases like the anatomical one that I showed, which are of healthy or even idealized specimens, but then how often are you going to see the diseased? What is the typical state of a diseased organ, for example? And so creating these type specimens is really crucial to the medical sciences. Atlas is obviously a visual media, and they define the visual standards which that the different sciences work with. And then finally, atlases, in some ways, and this is kind of an interesting and open question, define and organize communities of practice. So there are questions always about who can access and contribute to atlases. How are they formed? What kinds of committees go into making them? How are they agreed upon? And so they can and have had a kind of imperial function where one particular group wants to assert control over a discipline, and they create an atlas which becomes the standard. This is famous from all sorts of disciplines, and then you have competing atlases that come out that identify different standards. Obviously, in the age of the printed book, the cost and expense of production and circulation is a key factor. Now we have a different paradigm with the idea of open science, that everything is going to be open to everyone. So again, you have this. Another question comes in, which is who can contribute to that? Are there any patterns of exclusion that exist within open science, which would be a kind of paradox, but maybe it's the case. So the history of Atlas is a beautiful history, but it's also, I think, quite a provocative and interesting one to think with. And as I say, it's part one of this then 3-stage history of the Human Cell Atlas, which hopefully will be of use to Esther and Chris in making their film, of interest to members of the HCA and that community, and also maybe will give some kind of context and ability to interpret and situate the HCA for members of the public. Thanks.
Thank you so much, Boris. Incredible, mind spinning. If everyone's okay with that, we'll hold questions until all three of us have presented, and then open up to the audience for questions right at the end, because I think there will be lots of interconnections. So if you can make note of them, there will be lots of time for questions. But I'll hand over to Susie for an intermittent poll before we move on to Chris.
Thanks so much, Esther. And thank you so much, Boris. Oh my God, my mind is in fact blown. What a beautiful and, you know, so well considered and constructed presentation, just brimming with fabulous imagery. I'm just like saturated. Thank you so much. I've really enjoyed that. And so to our poll. I'm really interested, and we are really interested in who you are, and where you're coming from and why you're here, in terms of our audience. And so our question today is, in what capacity are you here today? Are you a speaker or an organizer like Esther, Chris and Boris, or are you coming in from a professional perspective, a non-professional perspective? Are you a HCA member? It would be really useful for you to share in what capacity you're here today, while we let Chris prepare for his presentation. Okay. How interesting. So we've got UK HCA members, we've got members from the arts and cultural community, we've got a variety of people from a variety of other professional capacities, and then a range of people coming in from their own interests. Well, you're all welcome, one and all. Thank you so very much for sharing that. And over to Esther to introduce Christopher Stewart.
Thanks so much, Susie. It's a great pleasure to introduce Christopher Stewart. Chris and I are collaborating on the film project, and Chris today will speak about the apparatus of looking. Chris is an artist and academic and researcher whose practice explores themes of surveillance, the invisible, secrecy, power, and masculinity. So Chris works across still and moving image, and his work has been exhibited internationally in galleries such as the Whitechapel here in the UK, National Museum of Photography, Open Eye in Liverpool, Photo Museum Winterthur in Switzerland, and his work is held in collections including the V&A. Curatorial research, writing, and image making are all central to Chris's practice. And I'll hand over to you now, Chris, to screen share. Thanks so much.
Okay, fabulous. Thanks, Esther. Let me just share my slides. Great. Okay, everybody. Hi, and thanks, Boris. Great. I may have to leave a little bit early, but I'm really interested in that relationship between your thinking on the HCA and the earlier work that you did in mass observation, which of course is a sort of fascinating empirical project with many pros and cons, of course, and its relationship to mapping and observation and hierarchy, I guess, fascinating. Okay, I'm going to actually just I'm going to stop my video so we just concentrate on the slides for my presentation. I'm going to whip through these pretty quickly. I've got a lot of slides, so I may sort of edit as I go. I did time it earlier, but it should be about 15 minutes, but I'll go quite quickly. Let me see. Okay. Great. Okay, so yeah, just to sort of reiterate, I guess, the presentation that I'm doing today really is fundamentally just showing fragments, I guess, of our research, the way we're coming at the HCA public engagement project and the film that Esther and I are making. Also a little bit about my own work. I think it's just useful to articulate that. And I guess as an artist, I've always been fascinated by the sort of hierarchies of looking, primarily in my earlier career. This tended to be around the sort of cultures of military and masculinity, some of the cultures that I came from, in a sense, myself, growing up within those cultures. I'm also an academic, as Esther says, who teaches histories and practices of photography. at the University of the Arts in London. So I'm fascinated by ideas of, mimesis really, or the idea of representation, rehearsal, similarity, simulation, resemblance, imitation, mimicry, etc. And of course, that's at the heart of my interest in the HCA really, the translation from one entity to another, in this case from tissue to data, as Esther sort of said previously. And I guess when Sarah was talking about the HCA years ago, what piqued my particular interest was more to do with that idea really of translation, when one thing becomes something else. So the film that we're making, or particularly my part of the film that we're making, is very much to do with looking at that translation really from one thing to another, from flesh to data, from analog to digital, if you like. So the film will be a metaphor for that. I'm going to begin with a quote, just really as a way to sort of link from Boris's talk. And it's a quote from the Borges story. So I'll just read it. In the empire, the craft of cartography attained such perfection that the map of a single province covered the space of an entire city, and the map of the empire itself an entire province. In the course of time, these extensive maps were found somehow wanting, and so the College of Cartographers evolved a map of the empire that was of the same scale as the empire and coincided with it point for point. Less attentive to the study of cartography, succeeding generations came to judge a map of such magnitude cumbersome and not without irreverence. They abandoned it to the rigors of sun and rain. And in the western deserts, tattered fragments of the map are still to be found sheltering an occasional beast or beggar. In the whole nation, no other relic is left of the discipline of geography. It's a quote that's also sort of infamously the beginning of Jean Baudrillard's book, Simulacra and Simulation from 1981, and his interest in all things simulated. And of course, the idea of maps and atlases are fascinating to artists, what we have here is a photograph of the Nimesini Atlas that was produced by Ebbe Warburg in the 1920s, started in 1924 and left uncompleted in 1929. And it placed the fragments of the past, in a sense, in conversation with one another, so to create a sort of linear picture of the connections to the present. So that idea of atlases being really informative to artists. I've got a few slides here just to sort of give you an idea of the kind of things that are influencing our research, but have been very important to me personally. This is a slide of Gerhard Richter's The Atlas Project, which is sort of a work in its own right, but also a series of images that he collects to inform his own painting throughout his career. But also to sort of try and make sense, I guess, of the sort of 20th century German history. Other artists, such as, here's a slide there of the Atlas Project in situ. Other artists using the idea of the Atlas are people like Penelope Enrique and her work, Sunsets, Portraits, taken from Flickr. This is from 2010. And so looking at that's a ubiquity of photography and the in this sense, the kind of cliche of the sun going down and how that's, that ubiquity is sort of formed and replicated. Works such as Patrick Pound's Portraits of the Wind from 2013. These are all photographs that he purchased on eBay and over many, many years, but to sort of try to curate this kind of, this kind of coherent sort of narrative out of this amazing sort of gamut of fragments that we can now find in the virtual world, but to bring them back together again to create a whole, if you like. And of course, the Carmen Winant series from My Birth, which I think Esther maybe will touch upon. I saw that slide in her presentation earlier, from 2018. Again, the idea of sort of the overdetermined idea of the presentation of images. And also the wonderful work, Gris Fatigue, by the French artist Camille Henrot, and the work that came out of her time at the Smithsonian Archives, looking at sort of origin stories, is a wonderful piece of work. So in terms of my own interest in ideas of some mimesis or likeness, of course, that comes primarily through the idea of photography and the history of photography. I teach histories of photography, and here we see the first photograph, or the one that still survives, really, from 1826, and that's Nice von Niepce's view from the window of Agras, which is fundamentally a picture taken out of his window of his courtyard. And it's the picture, in a sense, that we look back to think about the sort of birth of photography. Now, of course, Geoffrey Batchen talks about around about 22, the historian Geoffrey Batchen talks about around about 22 different versions of photography that were being developed in the 1830s. And of course, we know that photography was a discipline, in a sense, that sort of needed to happen. That idea of the sort of democratic form of representation that was required by the industrial middle classes at that point in time. And of course, the rapid development of photography in the 19th century goes hand in hand with the development of other sciences, and also the incorporation of photography into other sciences and pseudosciences, of course, notoriously, things like eugenics. Here we have an 1838 image, it's a daguerreotype produced by Louis-Jacques Daguerre, the official inventor of photography, of course, announced in January 1839. But here in 1838, we have this image, which is a wonderful image. It's the first known depiction of a human being within an image, and only caught because the protagonist here is having his shoes shined. And so the several minutes that it took to expose this photograph, anyone else in the photograph would have been blurred and would have disappeared. But we have this one protagonist who is caught for posterity. And of course, a different version of photography, Fox Talbot's 1835 lattice window. And there's a lovely story behind this. I was showing at the National Museum of Photography, Film and Television in Bradford quite a few years ago. And the curator, we had some spare time on our hands, and the curator asked if there's anything in the collection that we would love to see. I knew they had this image. It's a relic, of course, in terms of the history of photography. And so I asked to see it, and he laughed, and he took us down, a bunch of us, down to the archives, and opened this massive drawer. And in the middle of this massive sort of plan chest drawer was this tiny little white box with a white ribbon around it. And he said, there it is, but I can't open it. It's only got 20 minutes left. We only open it for Talbot scholars, and when we open the lid, we phone everybody up and give them the chance to come down and actually sort of set eyes upon this this little negative. And of course, Talbot's process was negative positive, unlike Daguerre and Niepce's, which were one-offs, and so that ushered in the era of infinite reproducibility in photography. And the rest is history, in a sense. Muybridge's 1878 image and the precursor, really, to film, the horse in motion, that showed for the first time a horse with all hoofs off the ground at the same time. And other experiments, such as Murray's short film from 1894, The Falling Cat, and also his pictures of figures in motion. And also there's an illustration there of Murray with his what is ostensibly a photo gun, depicting the figures in motion. And also the abstraction, in a sense, the experiments that then led to motion efficiency studies and the work of people like Frank Gilbreth. This is an image from 1914, and how photography was utilized to look at ideas of efficiency. And I sort of mentioned briefly there, photography's incorporation into sciences and pseudosciences in the 19th century, in particular, eugenicist practices, for example. But of course, photography, as art, but also as science, was crucial in terms of measuring the world and exposing the world to light up the sort of the dark corners, in a sense. And of course, that leads obviously directly for us in terms of our interest in the HCA, that sense of, what happens when we measure the world and we remove mystery, remove opacity, and create transparency. And some of the, obviously, the remarkable things that are associated with that, but also some of the issues, some of the cons perhaps of that also interest us. And here an X-ray diffraction image, so jumping to 1952, and this is an image made by Raymond Gosling, who was working as a graduate student under the guidance of Rosalind Franklin. So an image that contributed to as evidence to the sequencing of DNA, of course, the DNA molecule. And I've thrown Dolly the sheep in there as a sort of the ultimate kind of mimetic example, really. Can't leave Dolly the sheep out. And so also, the idea for us in terms of the film, really interesting to think about the translation of the analog, the idea of the analog into the digital, and also making those connections in a sense obliquely, I think, in terms of, I guess, what Richard Dawkins talks about in terms of, those particles, whether it's the gene or the byte or the atom, in a sense, the pixel for us, of course, in photography is a crucial part of the idea of looking and the hierarchy of looking. And here we see from 1880, the device produced by Shelford Bidwell, and where he had this remarkable idea: what if a picture could be broken into pure information and transmitted as an electronic signal? And so this is the telegraphic photography apparatus, where he achieved just that in 1880s, 1890s, early 1890. And we can see the first picture that was transmitted, the butterfly image here. And also, again, from Shelford Bidwell, this is around about 1890, the first image, one of the first images that was, translated from the analog to something electronic, with a sending machine and a receiving machine. And jumping forward about 60 years, the first digital image produced by Russell Kirsch of his son Walden, and created by scanning an analog photograph and producing a tiny digital file in 1957. So these histories are sort of fascinating for us. And a little bit more up to date, this wonderful picture here from 1992, which is the first photograph that was uploaded to the World Wide Web. And for those of you who don't know the history of this, it's a fascinating history. It's a photograph of CERN staff and a band that described themselves as the world's highest energy rock band. And it was produced by Silvano Di Gennaro, who was a computer physicist and the band's, inverted commas, manager. He scanned the photograph and saved it into his color Macintosh as a GIF file. And using a new editing program called Photoshop version 1, he cut the band away from the background and made this low quality promotion picture. And when Tim Berners-Lee walked into Gennaro's office, so the story goes, one day and saw him playing with the little digital photograph, Berners-Lee laughed and suggested that they make a website for the band on a little known computer network that was called the World Wide Web. And so Berners-Lee made a web page to advertise events taking place at CERN. And this was the first uploaded photograph of 120 pixels by 50 pixels. Because the web would have struggled with anything larger at that size, of course. And of course, the idea of digital manipulation, the infinite manipulation, in a sense, that we can now achieve through Photoshop and other software programs. And this is just the infamous image from a few years ago, 2008, of a doctored picture of one of the missiles failing to project, and the sort of doctored image on the left-hand side, and that need and that sort of desire, in a sense, to sort of attempt to decipher what's real and what's fake. Of course, I won't go into that in any more detail, but. And of course, to where we are today, this is a picture from Eric Kessel's project from 2012, Photography in Abundance, 1 million photographs taken from Flickr in a 24-hour period. And so that overabundance of photography, of course, is something that I'm particularly interested in. and obviously feeds into the some of the sort of more dystopian ideas of the development of technologies of looking and seeing. So to my work, as I said, I'm interested in, have been in terms of the histories of looking and the sort of hierarchies of looking, really. This is a project that I did on the southern Spanish coast, and it's part of the integrated external vigilance system down there. That was a 300 million euro wide surveillance system to curb immigration from Africa, which down in that part of Europe is at its nearest point to Europe, to Spain, of course. And so it was a project which fundamentally saw me walking along the coastline of Andalusia, photographing the evidence of these surveillance stations, which consisted of sort of surveillance stations, satellite imagery, personnel, and just looking really at how deeply surveillance and the idea of looking in that sense is now embedded in our society, how sort of mundane it's become, if you like, and how uncertain we are about some of those, or the evidence of some of those forms of looking. So these are just some of the images from that particular project. Surveillance stations, you can see Africa, Morocco, from across the Straits of Gibraltar there. Just sort of throw that in. It's a tourist company in Gibraltar called Parody Tours, which I particularly liked in terms of the idea of mimesis and the ideas of sort of the resemblance in the copy. Other projects that I've been involved with are an analysis really of the private security, private military worlds, particularly during the War on Terror. So I tracked and made contacts with private military companies all over the world. This is one group of military personnel hunting another group through the city of Miami, which I shadowed them for about 2 weeks. And they used the everyday landscapes of motels, shopping malls, bars, public parks in which to do their training. Many of them would then ship off to Iraq and Afghanistan. And so, yeah, just I guess, show a few slides that sort of, I guess, articulate my interest in ideas of looking, also particularly ideas of things like masculinity and how those things are constructed. This is a training camp in 2001, in the summer of 2001, training private military from different parts of the world. And again, that interest in the idea of looking and almost that sort of weaponized idea of looking. And also looked at training stations. This is called a kill house, which is an approximation of a domestic house used for training purposes. And these things have led to exhibitions. This is an exhibition that I curated with Esther called Staging Disorder for the University of the Arts. And this was an exhibition of 7 international artists, all of whom were interested in the idea of simulated spaces and geographies and architectures. And this is Brunberg and Shanarin at the end, which was part of their series called Chicago, which looked at the town of Chicago built by the Israeli Defense Force in the Negev Desert. And other artists, for example, Sarah Pickering here on the left-hand side, photographed the town of Denton, which is a Metropolitan Police training town built in North London. And then Claudio Hills's British Army bases that were built to train British troops before they went to Northern Ireland in the 80s, in particular 1980s and 1990s. This is the book that came out of that exhibition that we did. And then more latterly, I suppose, I've really got into the idea of CGI, and that's computer generated imagery, images that come from a source, an analog source, but then are translated, if you like, into something which is not analog, but is purely digital. And so for this particular project, for the HCA film, we're working with Liam Waters, who Esther and I have worked with before, a little bit in terms of CGI. So this is another exhibition that Esther and I curated, which was about the Stanley Kubrick archive and the 50th anniversary of Stanley Kubrick's 2001 A Space Odyssey. And it's an exhibition that we did with students, again for the LCC University of the Arts galleries. And it was very much about the idea of anticipation, about future architecture, and about how Kubrick in the 1960s worked with organizations and corporations like IBM and Hilton Hotels, actually, and got them to design what they thought would be future spaces in 2001. So the archive is very rich. And so we worked on this kind of exhibition with the students to look at that idea of sort of futurity and anticipation. And of course, in terms of sort of feeding into the idea of translation, the translation from analog to digital. Here are just a few sort of references, really. Tron, which was a sort of massive film when I was a kid, 1982, the Disney film Tron, which contains the idea of flesh being translated into data. There's a sort of scene within the film where Jeff Bridges, the main character, gets zapped and taken into the virtual world. And so, a film that sort of articulated all the concerns and fears about that sort of transition. And also a more recent, science fiction reference in terms of the HBO series Westworld and the opening credits to Westworld. So to the film itself, yes, we're working, obviously, looking at the spaces of the HCA, whether it's the computing, whether it's the sort of physical labs at Wellcome, at the Sanger, but also working with the Wellcome collection, the fascinating collection. So many wonderful images of the history of looking, and in particular history of sort of cell, the development of looking at cells. And of course, we've got Hook's image there in the top left-hand corner that Boris mentioned. And so many of the things we'll be looking at really in the film are, the apertures of looking and the history of that. But also the film at the moment, what we're thinking about is the film in two parts, one which is a very analog part, and then the second part being very much more of a sort of transition into CGI, again, as a way to sort of map and as a metaphor really for the HCA, the sort of material metaphor rather than a sort of direct one. I'm going to just end with a quote from Baudrillard, from something called Clone Story, from the publication I mentioned at the beginning. Of all the prothesis that mark the history of the body, the double is doubtless the oldest. But the double is precisely not a prothesis. It is an imaginary figure, which, just like the soul, the shadow, the mirror image, haunts the subject like his other, which makes it say that the subject is simultaneously itself and never resembles itself again, which haunts the subject like a subtle and always averted death. Thank you.
Thank you, Chris. That was absolutely amazing.
Speedy, yes.
Kind of wonderful, trajectory and genealogy of the evolution of photography and the ways of looking and seeing that move and translation from analog to digital. Really amazing. Thank you so much for that.
Fabulous, so much, and thanks. Thanks for the platform. I'm going to have to just nip out now to pick our little baby up, and but thanks so much. All right.
No problem at all. I'm going to introduce Esther Teichmann now, but before I do, I'd like to encourage and invite our audience to jump on their slido.com and do kind of think about any questions and upload any questions that you may have for our speakers. We'll be taking questions after Esther speaks. What I'll also say is that we are due to finish at 5:30. However, and we'll recognise that lots of people will have to drop off at that point. However, we may, if there's questions that warrant it, stay on after a few minutes after that point. So appreciate that you may have to leave, but you're also very welcome to stay should you like to. So to introduce Esther Teichmann, Esther's practice looks at the relationship between the maternal loss, desire, and the imagery, working across still and moving image installation. Recent solo museum shows include Heavy the Sea, Transformer Station, Cleveland and Monschwimmen, Rees Egelhorn Museum, Mannheim. Collaborations include Fantasy Photo Studio 2 with Monster Shetwinnet at John Hansard Gallery, and the co-creation and editing of the exhibition and book staging disorder with artist Christopher Stewart, who you've just heard from. Teichmann received an MA and a PhD in Fine Art from the Royal College of Art and is now head of programme of the Masters of Research and coordinator for Critical and Historical Studies at the RCA. I'm very, very pleased to welcome Esther to deliver her presentation.
Thank you so much, Susie, and thank you everyone for joining us. Chris and I are partners in life as well, so he's just gone to get the child, get the baby, and we'll come back for the Q&A. I'll keep my presentation really short to hopefully no more than kind of 10 minutes, and then 10-15 minutes. And then for anyone who can stay, we'll open up to questions and we'll have time for that. So I'm just going to go into screen share. Seeing the invisible, feeling the invisible. So today I'm going to be sharing fragments of my own work and other people's work and reading short extracts from mainly one book that's been a big influence in terms of my thinking on the film. Fragments on photography, invention, and motherhood. I just want to begin with a Haraway quote that maybe outlines my way of thinking about research, about connectivity and making relationships. Nobody lives everywhere. Everybody lives somewhere. Nothing is connected to everything, yet everything is connected to something. And a Foucault quote thinking about kind of montage, collage, taking apart what we have as given or as known and rethinking that. Knowledge is not for knowing, knowledge is for cutting. And I think those two quotes maybe are a useful sort of introduction to my approach to making and to research. So my relationship to photography is less connected to the idea of delivering transparency or mimesis that Chris just spoke of. So I'm not really interested in creating a copy, but for me, the camera and image function as metaphors for subjectivity, memory, and desire, the real being transformed from one thing into another. On a material as well as conceptual level, my work explores relationships between fiction, myth, and lived experience, examining the relationality of ourselves to one another, thinking about our bodies as sites of knowledge production. My work reimagines space and encounters through feminist subjectivity, exploring hybrid identities which are in between cultures and place, examining what this space of liquidity and multiplicity feels like. Both the written and visual components of my work utilize a mythologizing of the self, asking how we might write ourselves into our own narratives, and why this contextualizing of self within a fictional and imaginary realm is perhaps a necessary and powerful strategy. Central to the work lies an exploration of the origins of fantasy and desire and how these are bound to experiences of loss and representation. Our relationship to otherness, home, inherited grief, mourning, the maternal and familial, female pleasure, and the act of looking and being looked at are themes which are returned to across and through a layering of voices and visual approaches. And so I suppose this relationship to materiality, to the bodily, to flesh, to skin, the relationship to sisters, as mothers, mothers as lovers, the slippage of bodies is something that's present throughout my work and really has been from the beginning. But I'm also really interested in the materiality of the photographic. So I work with large format plate cameras. I work across digital analog, but that sort of physicality to wrestling with the image, wrestling with the technology, with the weight of the cameras, with the space of the darkroom is something that's really central to my process. And I'm interested in thinking about the kind of analogies between the photographic and the bodily. And this is a quote from Cadavan Cortes Roca from Reflections, Degree Zero Reflections on Roland Barthes' Camera Lucida. This play between light and skin, between the photograph and emanations can be registered in the French word for film, pellicule. From pellis, the skin, pellicule and film originally have the same meaning, a small or thin skin, a kind of membrane. So I'm interested in thinking about actually the photographic as this kind of surface in relationship to skin. The darkroom gives birth to the material photograph within the space of a red night, within a space of liquid, altered temporality, an unstable space of inverted, luminous non-fixity, a feminine maternal space. Like Abram and Torque's secret crypt of protected mourning, like the arms of the mother and lover, the darkroom is an enclosed space we enter, stepping from the light of day and a linear temporality into this other parallel modality of time. Here the past and present collapse into the futural. What seems about to appear and perhaps eclipse itself appears only to disappear, promising a refinding of that which has been lost, thus requiring a work of mourning at every moment. Bathed in the silent darkness of night, half-lit, half-light of red liquid, we see differently here. Blinded, there is clarity within the inverted hovering projection, within the floating, shivering movement of image appearing upon its material support. That's just an extract of my writing, thinking about the space of the darkroom. And just trying to move onwards. My computer's frozen for a moment. Apologies. Oh no. 1 second. Here we go. Sorry, my computer is totally frozen. Here we go, hold on. I'll move on. While my computer does this style of death, one of the first extracts I wanted to read to you is from Jessie Greengrass's novel Sight. It was published in 2018 and won the Wellcome Literature Award, what was the shortlist of the Wellcome Literature Award. And what's particularly moving in this book and really fascinated me is that it's this interweaving of the history of photography in relationship to science and medicine, alongside an account of the protagonist's relationship to motherhood and coming to terms with her own mother's death. And I just wanted to read a first extract that speaks about the first screening of moving image by the Lumiere brothers, coinciding temporally, historically, with the first or with the invention of the X-ray. The wheel of death is still going. Anyway. On the 28th of December, 1895, at the Salon Indien du Grand Café in Paris, the Lumiere brothers, Auguste and Louis, presented to the public for the first time a screening of a selection of their cinematographic films. Here we go. Oh, that's doing the whole thing. Oh, 1 second. Might just stop share for a moment. Okay. I'll read this. Well, it's not screen sharing. All that afternoon, along the Boulevard des Capucines, a line of people waited, their breath rising through the freezing air, in expectation of a wonder. Later, sat in rows on slat-back chairs, they saw it, the flickering black and white image of August holding his baby daughter up to a fishbowl, balancing the child on her feet so that she might look down at the water inside, the tumbling illusion of the film's frames making manifest inside the winter darkness, a months-old summer vacation. And at the same time, 600 miles away in the Bavarian city of Würzburg, Wilhelm Conrad Röntgen, chair of physics, ran through the streets to hand over a paper to the president of the university's Physical Medical Society, a first description of the X-ray. For weeks, while the Lumiere brothers had prepared their film, Röntgen, alone in his laboratory, its windows draped with heavy cloth to keep the winter's weakened sunlight out, had seen all that had been solid grow towards transparency. Opaque materials, wood, stone, his own flesh, had been reduced for him to shadowed outline, leaving the image of a substrate world spread out across a photographic plate, a catalogue of metal and bone, and all that would not rot to set against cinema's preservation of surface. I'm just going to go back and try the screen share. Backwards through. Here we go. So this is the first X-ray image. I'm just going to move on because I'm aware of time. So the second extract moves on to describe the making of this first X-ray image and its relationship to death and to love and to loss. At last, on the 22nd of December, 1895, Röntgen broke his run of solitude, the solitude of working in the lab. I jump forwards to a different section of the book. Returning home, he found his wife Berta, who for weeks had been his placeholder, moving through the routine business of their lives to keep the edges of it taut on his behalf, and he asked her to come with him. Without questioning, she put on her coat and gloves, and together they walked through the winter streets and then through the university, their footsteps echoing in its empty corridors, until at last they reached the door of his laboratory. It took 10 minutes for Röntgen to develop the picture, the only sounds his footsteps and the ticking of the regulator clock, which hung on the wall above them. And then it was done, and the lights on again, they looked at it. This picture, which has become Berta's enduring image, her skeletal open hand, fingers curved above the convex length of her palm. Across the lower phalange of her fourth finger, Berta's wedding ring is an uncompromising mark, its blackness against the shadow of her bones, a marker of the metal's immutability. Röntgen, who for weeks had been alone in his newly understood world, had sought with this image Berta's admittance to it, the making of the picture a gesture of both initiation and affection, the tenderness of her bones made visible to them, both, confirmation of the life which had formed such extraordinary structures, but these things are a matter of interpretation. To Berta, whose hands were solid, whose body unitary, who had not doubted those things that constituted her skin, her thoughts, the single object that was flesh housing mind, nor sought to understand them. It had a very chilly, soily smell of tombs. It is, she said, like seeing my own death. She turned away and refused to look again. So I was interested in terms of thinking about invention and the photographic and the overlaps between the domestic, the medical, the scientific. Here I just want to show extracts of references of what we're looking at the moment. In the bottom right, a moving image piece from the Wellcome collection that shows cell multiplication in gestation. And the top left, Stan Brackage's Window Water Baby from 1958, I think, of his wife giving birth. And I've been reading to understand what Sarah's doing because I don't. Like, funny music goes off in my head when she starts speaking about it. You can see genetics and how different our brains work. But this is a very useful book, The Making of You. It's a recent book published by Wellcome, A Journey from Cell to Human by Katharina Wester. And it describes from the position of the cell, in a sense, our origin stories. I just want to read one section from the chapter called The Invasion. It is one day after your conception, and the small hairs in the fallopian tube are nudging the tiny round cell down the canal, slowly, carefully. On the outside, everything looks perfectly calm. Deep within the cell's interior, however, a sophisticated mechanism is working tirelessly, creating precise copies of your DNA molecules. Soon, each chromosome becomes an X shape formed by two identical DNA molecules attached at the center. The chromosomes assemble at the center of the round cell, row upon row. At the same time, the cell spins a web of protein from either side, and long, thin strands reach towards the center, taking hold of the chromosomes. The cell then stretches and becomes elongated, while the strands pull the DNA copies towards each pole. It's beautiful to observe under the microscope, like a tiny firework. Finally, after about a day, it pinches in the middle and splits into two. And it continues like that. The cells copy and divide, copy and divide. And we've been thinking about the different chapters in the Human Cell Atlas and really taking the developmental cell atlas as the origin, really, as kind of the central thread, as one of the central threads in our film, thinking about that as kind of the origin of us. But I'm really interested in thinking about actually what makes up the developmental cell atlas and that relationship to donation and gifting, and thinking about, and here I'm kind of speaking freely without script, but in talking to Sarah and trying to understand actually where the cells come from for all of the different parts of the HCA, but especially for the developmental cell atlas, I was really struck by the that section of the HCA being made up of tissue donated from women from abortions primarily, and thinking about abortions where in the kind of first 5 months. And so I was thinking about the history of sonography, the history of gestation, the decisions always complex, never easy in terminating a pregnancy, and thinking about the complexity around seeing and kind of transparency. And Jenny Bangham, a writer and who will also be working with us, gave me the reference to this amazing essay, book called by Lisa Mitchell called Baby's First Picture, that looks at the ultrasound and the kind of politics around fetal subjects and imaging, and how actually the sonogram and the history of the ultrasound is a really complex one in terms of, in a sense, also differentiating the fetus from the mother, right? And therefore also, in some ways, taking away from women's rights in relationship to reproduction rights, and kind of in place of the rights of the fetus. So I was really interested in the kind of complexities around that, and the relationship to kind of loss, but also of donation and gifting in relationship to the developmental cell atlas. So that sense of making this really difficult, I'm sure always difficult decision, that right now is not the right time or context to become a mother, for always individual reasons. But then to gift that tissue towards science. So I think that duality was really fascinating to me about this chapter of the HCA, which is maybe quite different to the other sections. And here, this slide shows just an extract. I won't read that quote from the book, which is brilliant, really interesting. And also some images of a collaboration between Tracy Emin and Louise Bourgeois, as well as Emin's early work, Something's Terribly Wrong from 1995, based on her own abortions. And in the bottom is new work by the photographer Rinko Kawuchi's amazing new book called As It Is, that combines images of her child and birth and writings. But the images here, the medical images, show some of the first sonograms used and kind of championed by Ian Donaldson, and in the 1950s. And he was based he began medical at St. Thomas's here in London, and then worked at the University in Glasgow. And he pioneered the use of ultrasound to measure fetal development. But he also became known for opposing abortion, and campaigned against the 1967 Abortion Act. And so I was interested in that kind of complexity of invention and kind of politics, and ethics around kind of ownership, who owns, what in terms of our bodies. And so I just want to close, because I'm aware of time, with a last, this is Carmen Wynant's amazing work, My Birth, that Chris touched upon as well. And I just want to close with a last section from the novel site that talks about sonography. At last, the sonographer stood up. For a moment, she fiddled with the large machine beside the bed, angling its articulated monitor, then saying, "This will feel a little cold." Squeezed gel onto my stomach, a great chilly splurt, which I would afterwards be left to wipe off with a paper towel. My furtive embarrassment at the task, the first in a series of slight indignities, which over the next 6 months would strip me, layer by layer, until at last I was nothing but flesh, and would lie naked in another room and scream while strangers came and went about me. The sonographer passed the ultrasound's transducer backwards and forwards, pressing down until I winced, staring across my shoulder at the monitor, which Johannes and I were not permitted the sight of. I watched instead her face, the small frown of concentration that lay in the ridged skin between her eyes, and tried to force myself to some understanding of what we had to lose. The night before, Johannes and I had sat side by side on the sofa and, in half-made sentences, like tendrils, cautiously unfurling into dangerous territory, discussing what we might do, without either of us being able to quite articulate what it was we spoke about. And I don't know, I said, how I might feel. It would be dreadful. Meaning all the time that I knew what our decision would be, but I didn't know what degree of guilt or distress I might feel, all outcomes seeming to me so far entirely hypothetical. And I was worried what I would feel, that I would feel nothing for this entity, which was as yet more idea than child, which was in its own presumptive wellness experienced as the expectation of an unimaginably different future, and as a combination of sickness and obligation, a requirement to regard my choices as circumscribed. And I'll leave it there, but I was really interested in that section that really addressed, you know, the kind of contemporary relationship to maybe also thinking about the normality, you know, experience that myself in terms of doing really early DNA tests in pregnancy, and then thinking about the decisions that we would make in relationship to whether we would terminate or continue with the pregnancy, and thinking about kind of the unspoken complexities around that in terms of guilt and social taboo. And I think this novel kind of touches on these elements of invention, photography, science in incredible ways. And so here, kind of a different version of mood board, reference board that Chris pulled up, that is our the starting point of the film that we're really just in the first days of. So I'll close there because I'm aware we're out of time, over time, and want to leave time for questions. So please do, pose questions to the panel, and Susie will do a short poll to close the session.
Fantastic. Thank you so much, Esther. While you're, I see Charlotte's on it. She's got the poll just on the go. And while she's kind of bringing that up, let's just, so what 3 words come to mind having seen our presentations to help kick us off in terms of some questions? But from my side of things, Esther, just to say that, you know, what a beautiful, you know, way to introduce the poetic, to introduce the emotive, the subjective, the experiential, you know, all of those aspects that envelope the science, the mechanics, the materiality, the tools, the inventions of seeing and looking and imaging through the Human Cell Atlas. I have to say that was just a really, it's been a visual feast over the last hour and a half. I've been completely overwhelmed by how beautiful these presentations have been. Thank you so much. Really wonderful. We have vision as a word, which is a really interesting one. I would definitely suggest that that is a potent word to come out of these presentations. We have confusion, perspective, detachment, art. How wonderful. Creating imaging, thinky. These are really interesting. Structure, beauty. I'm going to let this roll just for a couple of minutes more just to see what else emerges. Please do share your thoughts. Proliferation, biosocial, light, scale, beauty. Wonderful. I realize my vision is not so great. These are quite small on my screen. Fantastic. We have a selection of really interesting words. How can I pose the first question to the panel? Maybe Boris and Esther. Maybe if Christopher's back, I'm sure he'll jump in if he can. Are there any of these words that have been posed by our audiences that particularly resonate? You're doing what I'm doing, you're peering in. Please peer in and maybe identify some of these words that maybe resonate particularly with you, and maybe kind of riff off them and give them response.
I guess that relationship to kind of differing perspectives and thinking about, I mean, as said, we're in the very, very early days of starting to explore and starting to have discussions with Boris, which every time open up just the most amazing frame of references. For example, the Cloud Atlas, which I had known nothing about, and then thinking about the analogies to kind of the digital cloud and open access data and the HCA kind of structure. So I think that sense of kind of interconnections and layerings and thinking about different perspectives and histories in a way, yeah, the multiple.
Absolutely.
I'm just reading the words. That's not answering. Yeah.
We have a question. I'm conscious of time that it's 22. So we have a question from the audience. So Chris Stewart mentioned the relationship between eugenics and photography. Please expand on this troubling historical intersection and recommend further reading.
So I think Chris is just getting the baby, but I can answer to that too, because come from a history of photography as well. In terms of kind of the best further reading, I'll get back to you on that. But I think Chris was pointing to the, that we can't deny the history of photography being one that is hugely problematic in relationship to kind of measuring and defining and categorizing difference, right? Be it colonial kind of histories of saying this is the primitive other and here are the measurements, and using that to justify horrific kind of political actions, or in terms of kind of medical histories, actually, in relationship to women's bodies, in relationship, we think about the images in Salpêtrière of hysterics, for example, the kind of very uncomfortable history that photography has in relationship to documenting and measuring, and therefore using kind of the other and that distancing as a way to justify, really in a way, criminal acts in all different ways. So I think that the idea of, oh, this is what, if we layer these images, this is what a criminal looks like, this is what an insane person looks like, right? So that history of photography is there, and is something that as artists who work with photography, as academics who teach the photographic, is always something that we have to include and make kind of people who are studying the photography and working with the medium aware of and understand. So I think the origins of photography, whilst kind of multiple and visually in many ways incredibly exciting and interesting, is also very, very problematic. Yeah, does that answer that? Maybe there's lots and lots of reading on that. Really every book that looks at the history of photography would touch upon that, but I'll get back to you about kind of key texts if I can, if you give Susie your contact.
Please do, please share your contact. So we have a second question, which I'm really interested, maybe potentially pitching to Boris, if that's okay. We heard a lot about science inspiring art, but what about art inspiring science? A very useful question to ask.
Yeah, that's a good one. That's a really good one. I mean, it's maybe a little bit easier if I just go sort of straight back to the earliest material that I was showing, and there it's really not possible to draw a very clear line between the two, which it was a bit suggestive, but that's why I showed the Rubens illustration of the atlas with the celestial sphere on his back casting the shadows, which illustrates projection. And that's one very easy example where the laws of projective geometry are established and published and theorized as much by artists as by people we would identify as scientists. So one of the main books on geometrical optics is by Albrecht Dürer, who's obviously much more famous in art history than he is in the history of science, although there's a kind of slippage there. And I suppose if you were to think, maybe if you stick with fine art, it's difficult, but if you think of illustration, engraving, reproducing images and those practices, you can very easily, especially thinking about atlases and these kind of printed encyclopedias or collections of images, then the history is really a history of both things. So the history of clouds is another excellent example, where even if you think of the first person who named the clouds, Luke Howard, he trained himself to become a watercolorist who was able to draw or illustrate the cloud types. And he struggled with this his whole career. And in fact enlisted artists to help him draw these scenes, which would then become the type specimens of the clouds. So there's the minute you get into this question of communication or reproduction, classification, these are aesthetic questions as well. Then that's obviously true in probably all of the examples I gave. And there's always a close relationship between image making and the arts, and then image making and the sciences. So I don't know if the influence question is kind of hard, because we're not in a sense talking about scientific discovery or innovation or development. We're talking about communication and representation. But once you start to look at those sorts of practices, it's really they're really two sides of the same coin, at least through to the kind of 20th century.
And I think that separation, maybe to kind of follow on from that separation of thinking about art and science as separate, really, or kind of thinking about disciplines in that way is quite a recent kind of separation. So when we think about Hooke, for example, as, was also a draftsman, amazing drawings, right? Worked with architecture, astronomy, built kind of early microscopes. So I think that separation of thinking about scientific invention and creativity within the arts as kind of separate from one another, that's only a fairly recent kind of siloing of disciplines that I think in some ways, whilst of course still totally there, is also beginning to break down in interesting ways, and that working across disciplines, even if they still be within kind of scientific or medical fields, or whether in the widest sense within art and design, that working across disciplines is becoming, I think, much more normal again. And thinking about the kind of productive nature of interdisciplinarity and kind of collaborative research, I think is definitely very much in the ether.
I agree, and I think that's a really interesting point, Esther. I think this separation between arts and science can be kind of traced back to kind of 1959 C.P. Snow, his kind of two cultures kind of read lecture. I think it was Cambridge, wasn't it, that he kind of did that lecture, which obviously at the same time, you were having experiments in art and technology happening in the States. You were having Leonardo Journal kind of kicking off kind of activities in Europe. So you kind of had this kind of provocation almost of a separation of those two cultures, but actually quite rapidly a kind of a challenge to that paradigm and perspective, which I've been looking at more recently myself in relation to this project, which is really interesting. And I think just more recently in terms of that relationship between kind of art and science and the kind of influence on science, and pointillism is something that one of the HCA members, Isaac Go, mentioned to me in relation to the kind of evolution of kind of visualizing distributed data sets, which is just a really kind of direct and wonderful example, I think, in terms of an artistic methodology, kind of really informing methodologies across the sciences. So that was really interesting. And then a final question, and because I'm conscious of time, and thank you so much for those who have stayed with us. Islamic arts-led science in terms of geometry to architecture, pyramids, also Da Vinci, of course, wonderful examples. Thank you so much for whoever anonymous is for those.
I think there, if I can also just say, I think I'm really interested in the parallels, maybe not so much where art inspires science or science inspires art, thinking kind of in such binary ways potentially, but actually thinking about the parallels in terms of invention and creativity, and that relationship to not knowing, that relationship to the imagination, the kind of imaginary leaps, right? Having to envision something that does not yet exist, but thinking, kind of working on a hunch, on maybe kind of a guttural feeling, and thinking about those intersections and parallels, between kind of processes of making within kind of creative production and within scientific research. Because I think there are a lot of overlaps there in terms of how we think about what it is to imagine something into existence.
I totally agree. And just I think in lots of ways, the HCA and projects in relation to yours, Esther and Chris and Boris, it feels very much like, people are kind of pushing beyond the kind of edges of their methodologies in many respects, and connecting in really potent and interesting ways in that space between. So we have absolutely two very similar cultures. And Sarah, do notions of you of I can't pronounce.
I don't think Sarah. I think Sarah's with the kids at hockey now. She's had to leave, but maybe some of the other HCA scientists can answer that. I'm sure. Maybe I can't.
We can certainly take this to Sarah.
Yeah. No problem. I will screenshot that and ask her.
Most scientists I know are obsessed with art. Absolutely. Fantastic. And I think on that note, that might be a good way to wrap up this fascinating session. I'd just like to thank every single one of our speakers once again. Boris Jardine, Christopher Stewart and Esther Teichmann. Sarah, I know this is being recorded, so for posterity, thank you very much for your wonderful discussion that kicked off this session. I'd absolutely love to give my thanks to our HCA team, Paul Gibson, Charlotte Bathram and Tracy, who are backstage making all this wonderful stuff happen. But most importantly, I'd really love to thank you, our audiences, for coming and sharing your thoughts and your questions and your ears with us to hear more about the story of the Human Cell Atlas by Esther Teichmann and Christopher Stewart. Thanks again. I'll leave that go now and say goodbye. Take care.